Yep.
Cool. Okay. So thank you everybody for having me. I'm really excited to be here. And I'm so pleased that so many of you have showed up tonight. I hear it's an unusually large audience, so thank you all for being here. If you were here in the April meeting, for the April meeting, Dr. Bridget Clarkson's meeting, I think she set me up quite nicely for my presentation. She talked about everything on the algae side of the intertidal, and today I'll be focusing on everything invertebrate related in the intertidal zone. So she gave me quite a nice introduction to my presentation. For my presentation, we're going to start with some introductions and definitions just so that everybody's on the same page and we're all talking about the same thing. And then I'll talk about why the intertidal is unique and why I like it and why I study it. We're going to do a who's who. So next time you're out paddling and you look at the intertidal, you can actually ID what you're seeing and some fun facts about everything. And I'm going to talk about my research that I do at the Department of Zoology. And I'm going to pepper the entire presentation with some of the other research that's going on in the lab that I don't do, but that I hear a lot about on a daily basis. And then we'll finish with how you can get involved as paddlers and kayakers. So my mom kind of covered it. Besides her daughter, who am I? Currently, I'm a second year master's student and a graduate student at the University of British Columbia, and I'm a master's candidate in the Department of Zoology, specifically in the Harley Lab. Before I decided to go back to school, I worked as a fisheries biologist for a small consulting company located mainly out of Port Hardy in northern Vancouver Island, and then I decided to return to school. Work covered from the BC coast from Haida Gwaii to Victoria, and it centered around aquatic ecology and resource management. I did a bit of a 180 to end up in the ocean, though. Before that, I completed an undergraduate degree in the Department of Forest and Conservation Sciences at UBC in their Faculty of Forestry. So I've covered terrestrial ecosystems, and now I'm into the aquatic ecosystems. So I said I was a member of the Harley Lab at UBC. What exactly is that? It's supervised by Dr. Chris Harley. That's him. If you're on the shores of Vancouver, you might see him around. He always wears the same hat. Look for the purple hat. It focuses on coastal marine ecology and the impacts of climate change. More specifically, we study changes in temperature, we study ocean acidification, which is what I study, altered salinity, invasive species, and long-term ecological change. Where, in a nutshell, we just study how climate change is impacting the intertidal zones and the animals that live there. If you're interested about this kind of thing, you can find us online at zoology.ubc.ca/the Harley Lab for up-to-date information on who's studying what in the lab. And you can periodically, not very periodically, find us on Instagram at Tides of Change. We are Tides of Change because apparently there's already a Labrador Retriever who is named Harley, who has his own Instagram account. So we are Tides of Change. So because I spend so much time in this lab, much of this talk will focus on science, um, but hopefully not all of it. So what exactly is the intertidal zone? Simply put, it's where the ocean meets land between the high tide line and the low tide line. However, the world is incredibly varied, and where ocean meets land varies hugely across the landscape. So your intertidal zone is going to look different no matter where you are in the world. So estuaries are one type of intertidal zone. They're an extremely unique interface between saltwater and freshwater that you don't really find anywhere else. Mudflats are also a type of intertidal zone. They look barren and boring, but they are deceivingly productive and they're teeming with life below the surface. And you just have to dig an inch and you'll find so much life. Beaches are a common intertidal zone, but they can also abut sand dunes, which is a very uncommon intertidal zone. They comprise 1% of BC's coast, and it's one of Canada's rarest ecosystems. So it's extra special to find sand dunes. But my favorite type of intertidal zone, and the one that much of this talk is going to center on, is the rocky intertidal. It's what people think of when they say tide pools, and it's basically anywhere on the shore that's covered in rocks or bedrock. I could have also included eelgrass beds or salt marshes on this slide. They're all types of intertidal zones, but I didn't have any pictures of those, so I didn't include them. The Rocky Intertidal is my favorite of all those intertidal zones. It's also considered one of the harshest environments in the ocean. It's a really, really difficult place to live for all organisms that live there. Most animals in the world are adapted to a very narrow range of environmental conditions. So you're not going to find a camel in the Arctic and you're not going to find a polar bear in the desert because each of those animals is adapted to either a hot environment or a cold environment. But in the Rocky Intertidal, you have to be adapted to both. It's hot, it's cold, it's everything, and you can't have a very narrow range of environmental conditions. And this is because the tide is always changing. I know as kayakers, you're intimately familiar with the tides, and they change everything about this environment every 6 to 12 hours. So it's always changing. It has many extremes. There are large periods of time without water, so you're at the mercy of the air temperature and the air environment, but then you're also submerged and you're at the mercy of whatever's going on in the ocean. So there's large temperature, moisture, and salinity variation. And on top of that, you're also susceptible to high wave and wind action, erosion. You can get tossed around. It's a really difficult place to live. So the intertidal zone technically is split into 4 subzones. So there's the spray zone, which is just where like sea spray might, it's not technically an intertidal zone, but it's where you might get sprayed with salt water. And then it's split into the high, mid, and low intertidal zone. So the high intertidal is anywhere, is the area of the intertidal that's submerged for the least amount of time. So it's the area that's going to be closest to the shore. It's typically populated with species that can withstand extended periods not covered in seawater. So they're not going to what we call desiccate. They're not going to dry out and evaporate when there's no seawater on top of them. This tends to be organisms that are able to store their own water, such as barnacles, mussels, or oysters. So these are things that are open and feeding at high tide when they're covered with water. And then when the tide goes out, they snap their shells shut and they keep seawater in there to keep their tissues wet so that they don't desiccate. So if you're ever kayaking and you pick up an oyster and you shuck it on the beach, it's going to look dry. And then when you open it, it's got that salty seawater inside of it, and that's just to keep it alive during low tide. Conversely, the low intertidal is the exact opposite. It's not such a harsh place to live. It's not so unforgiving of an environment because it's buffered by seawater. It's covered by the ocean for more time of the day than the high intertidal. So organisms don't have to be quite as hardy to live here, which means that it's highly biodiverse and tide pools are going to look more like the picture on the left. Lots of different species, typically things that are not adapted to being dry at all. So they're going to be your soft-bodied organisms with a high water content, such as anemones, sea stars, nudibranchs, algae, etc., etc., etc. So we talked about the tides. They have a huge impact on the intertidal zone, and especially in Vancouver and the surrounding area. So this is Point Atkinson data that I'm going to show. It's a tide chart. This is July tides for this year, 2025. And you'll notice that in Vancouver and nearby areas, the low lows for the year happen to be in the summer around midday. So organisms that are exposed at low tide are exposed to air temperatures, and they're also exposed to air temperatures when they're going to be the hottest, so in the middle part of the day during the hottest part of the year in the summer. So this means that there's huge extremes in the summer, very hot temperatures, and it's the exact opposite in the winter. So our low lows in the winter are typically in the evening or in the middle of the night. So that means that organisms not only have to withstand the air temperature, they have to withstand those like extreme temperature extremes. This, however, is not the case anywhere else. So Vancouver is just drew the short straw. This does not occur on the west coast of Vancouver Island, and that means that you get more productive beaches. So Botanical Beach near Port Renfrew, that's why it can be so productive is because it doesn't experience these crazy temperature extremes. When those tides perfectly line up with the weather, you can get deadly heat domes and cold snaps. So this is Vancouver Harbor data. In 2021, temperatures reached 34 degrees Celsius and were above 30 degrees for 4 days in a row. So this is extremely unusual for Vancouver. We are not a hot city. We do not experience 35-degree weather for a prolonged amount of time. So we called that a heat dome. But it was even worse for intertidal invertebrates. So our air temperature was 34 degrees, but our black rocks in the intertidal are going to heat up faster. So they measured temperatures anywhere near 50 degrees Celsius in the intertidal during this heat dome. And that means that sessile organisms, so organisms that can't move around, can't hide, can't find any refuge from that sun, have nowhere to go. So it resulted in a 100-meter stretch of beach in 10 billion barnacle deaths that year. Conversely, last year in January, we had a cold snap. So 10.6, minus 10.6 degrees, and we were below freezing for 7 days in a row. And that lined up with when our low tides were in the middle of the night. So that was also a temperature and weather anomaly. And it provided a great opportunity for Sojung Park, who is a PhD student in our lab, to study this. So she is doing a qualitative analysis of this cold snap. She's trying to figure out where and what organisms died during this cold snap and whether it's related to where you find them in the intertidal. So our high intertidal organisms more freeze tolerant because they spend more time outside of the water versus low intertidal organisms. So that's what she's working on these days. So with all these heat domes and cold snaps and temperature anomalies and climate change, enter Chris Harley. He's my supervisor, and his work really came to the forefront during the 2021 heat dome. Residents and neighbors of the beach started reporting that like the smell of death was very apparent, and it started making the news. So you may have heard Chris Harley in just some of the CBC News articles that came out of this heat dome. He was everywhere for about a month. This is where I first heard of Chris Harley, and it's when I decided that I wanted to do a graduate degree and I wanted to do it with him. He's studying how these heat domes are affecting the intertidal zone, and that 10 billion barnacle number comes from his research. So he studies and his lab studies adaptations and how climate change is affecting the intertidal zone. So with these temperature extremes, organisms have to be able to not cook in the heat or freeze in the cold. So it's a big ask for tiny organisms. And to do that, one of the ways they control this is by creating ice binding proteins. So these are proteins that they have in their body that allow them to maintain liquid water in their cells, even when their body temperature is below zero. So you might have really cold temperatures, really cold air, but you can still stay alive because your water in your cells hasn't frozen yet. Conversely, you can also make heat shock proteins. So this allows organisms to adapt to and withstand hotter temperatures. So this is where Natalie Rivlin, she's also a PhD student in our lab. This is what she studies. She doesn't study heat shock proteins themselves, but she studies muscles. So she studies how muscles might do better under climate change scenarios if they're subjected to repetitive, moderate heat stress. So in the same way that you might, if you're training for a marathon, you might run multiple little runs to train. Muscles would prefer to have several not so hot days in a row before a heat dome, and then they'll do better in that heat dome. They're less likely to die. So this is going to be great news for climate change. However, eventually, all organisms do have a breaking point, and they will eventually die. So we're going to pivot a little bit now. We've talked about the intertidal zone, so we'll do a quick, well, not so quick, but some who's who. Who are you likely to see in the intertidal zone? I would say the number one thing you're going to see or you're going to notice are the ochre stars, Pyosaster ochraceus. They are vibrant, they're colorful, they're beautiful, and they're very, very common. They are the most common species of sea star that you're going to find on the coast. um, they are also a keystone species. So a keystone species is any species that has a disproportionate impact on the environment. So if you take it out of the environment, that ecosystem is going to change drastically. It's not going to be the same ecosystem that it was when it was there. So Piesaster have a huge impact on the intertidal zone. Um, Piesaster was also absolutely annihilated by sea star wasting disease in 2013. So if you've been kayaking for a while and you remember 2013, 2014, 2015, you might not have seen these at all. They got absolutely decimated by this disease. But it's made an incredible comeback, especially in BC. There's a lot of debate about what recovered looks like for an environment because we don't have any data from before the outbreak of the disease. But I think scientists are pretty happy to say that it's recovered, at least in BC. It exists in two main colors, which are likely associated with their diet, so purple and orange. And this was an undergraduate project that started last year to see if you feed purple sea stars the orange diet, can you turn them orange? Or if you feed orange sea stars the purple diet, can you turn them purple? And the answer is probably, yes. We would need some longer term studies to say for sure, but it's really interesting. And we'll touch on it a little bit later when we talk about mussels, but really interesting. So I said ochre stars got annihilated by sea star wasting disease, but what exactly is that? So it's a disease that spread from Mexico to Alaska, and it affected about half of our species of sea stars on the coast. It's very severe, and it caused 90 to 95% mortality in some areas, 100% for some species. Causes lesions, loss of limbs, and eventually death of the organism, and it's very, very quick. So from experiencing no symptoms to dying can be anywhere from a few days to a week to maximum 2 weeks. An early warning sign is twisted arms. So if you see a sea star that's not kind of like if you find them under rocks, they're going to be in weird positions, but this one is out in the open, but its arms are still twisted. That's a very early warning sign that it's a sick sea star. And then it develops these white spots, white lesions on one or two of their arms, and then it spreads, and eventually they lose a limb or they just kind of disintegrate into goo. The exact cause of this is as of yet unknown. The latest published guess is that it's a virus. We're still working on finding out what exactly it is. It's a big marine mystery, but we do know that it's exacerbated by warmer temperatures. So although the epidemic is over and things are recovered, or some things are recovered, we still get blips in the summer when the water temperature warms up. There are little tiny outbreaks of sea star wasting still. One organism, the sunflower star, Pycnopodia helianthoides. This one used to be found from Mexico to Alaska, and it was especially hard hit by sea star wasting. So almost 6 billion individuals died in 3 years. No one really knows why this one was hit so much harder than everything else, but it hasn't really recovered in most areas. So populations have declined by 90%, and they're pretty much extinct from the southern end of their range. You can't find them in warmer waters in Mexico and California anymore. In BC, we're really lucky to still have a viable population. It's isolated in pockets in fjords and random locations in cold water environments. These two juvenile pictures I took in Port Hardy, so pretty far north. It's now listed as critically endangered by the IUCN, which is the International Union for Conservation of Nature, and it's very closely monitored. And if you see one, if you're out kayaking and you see one of these, all the scientists want to know. So you can record your sighting and they'll track the outbreak of sea star wasting and the recovery of sunflower stars from citizen science observations. So we'll come back to this at the end of the talk, but if you see one, we want to know. Some other sea stars you might find, mottled stars of Asterias, are also highly affected by sea star wasting. They're in the bottom left. They're more of a subtidal species, so you're less likely to see them walking on the shore, but you can definitely see them from a kayak, and you can see them from the intertidal. Some stars are naturally more resistant. So leather stars, dermasterias, bat stars, blood stars are all just naturally resistant to sea star wasting, and their populations have done quite well despite the outbreak. They're mysterious. The leather stars in particular are really easy to see in the intertidal, but they're beautiful. Okay, these are my favorite organisms that you might come across. Nudibranchs, naked gills. It means naked gills in Greek, I think, or Latin. They are sea slugs, and they come in two main groups, the dorids and the aeolids, and it's defined by how they breathe. So if they have tufts on their back, like I don't know if you can see my pointer, but if they've got tufts of gills on their back, then you're a dorid. And if you have those feather-like serrata, then you're an aeolid. They have unique defense mechanisms. So these are absolutely crazy animals. They come in bright colors, and this warns of toxins and bad tasting chemicals. The hooded nudibranch in particular, the legend says that if you take them out of the water, they smell like watermelon. I have not found one in the wild, but if you come across a hooded nudibranch, take it out of the water and smell it because I want to know if this is true. They don't produce their own defense compounds, but they absorb them through their diet. So some nudibranchs eat sponges or corals that contain bad tasting chemicals. Some eat anemones or jellyfish and ingest their stinging cells, the nematocysts. And then instead of stinging the nudibranch, they absorb them and then use them to sting their own predators. Some eat corals that contain a photosynthetic algae that allow the nudibranch to become photosynthetic itself, and then it doesn't have to eat for several months. And this is one of the only cases of a photosynthetic animal, so very unusual. And in particular, latrunculin A is a highly toxic compound that's found in some sponges, and it's also highly toxic to cancer cells. So scientists are researching nudibranchs for possible cancer treatments. I also noticed that there was a few people here from Nanaimo, Cheryl from Nanaimo. You have your own nudibranch on this slide, the Nanaimo dorid. This was first described by Charles O'Donoghue in 1921 in Nanaimo in Hammond Bay. It also goes by an American name, but that dates to 5 years later. So we're going to call it by the Nanaimo dorid.
And it's going to cure everybody of cancer in Nanaimo.
Maybe, maybe. There's also green surf anemones. So Anthopleura xanthogrammica, these are the really common ones that you'll see in most tide pools. They're bright, vibrant green color. They're also usually very, very large, like very large. They're beautiful. They are not green by themselves, though. They get most of their color from a photosynthetic alga that lives in their tentacles. So they have a symbiotic relationship with this alga. However, in stressful conditions, this relationship is interrupted and it can cause anemone bleaching. So this is very similar to what's going on in the Great Barrier Reef in Australia and across the world with coral reef bleaching, where stressful conditions, usually extreme temperature, but not always, stressful conditions cause the anemone or the coral to kick out that symbiont, in this case, an algae. And then the anemone doesn't survive as well without it. It doesn't have that same green color. It doesn't get the same nutrients, and it ends up looking something like this. This is not a green surf anemone because I didn't have a picture of that, but this is a bleached anemone, and they look this white in color. This is something Sophie Johnston in my lab studies. She is a PhD student. She is the queen of the symbionts in my lab, and she studies green surf anemone bleaching. Northern abalone. These are also one of my favorite organisms, and they're usually really overlooked, and I don't know why because they're such a cool organism. It's a culturally significant species. So the shells are traditionally used for First Nations art and clothing, and the meat is a delicacy to First Nations across the coast. However, at one time there was a commercial fishery for abalone, and it was significantly overharvested. And between 1978 and 1990, populations declined by over 80%. So this led the Department of Fisheries and Oceans to deem harvest illegal in Canada since 1990. 10 years later, in 2000, it still had not recovered. Populations weren't looking any better, so it was listed as endangered by COSEWIC, which is the Committee on the Status of Endangered Wildlife in Canada. It's like the IUCN that listed the sunflower star, but it's just for Canada. So it's national. Um, 3 years later, after it was listed by COSEWIC, it wasn't looking any better still. So it was federally protected in Canada under the Species at Risk Act. So this comes with legislation and federal protection for the species, and it has been listed ever since. In 2006, it still wasn't looking any better. So we're now 16 years past that DFO fisheries closure, and it's still not looking too good. So the IUCN stepped in and they also listed it as endangered. So in 2006, it was listed nationally, it was listed internationally, and it was federally protected in Canada.
And the,
Now,
and the
10
First
years
Nations,
later,
can the
sorry?
First Nations, can First Nations harvest them still?
No. They closed the commercial fishery, they closed the recreational fishery, and they closed the ceremonial fishery and cultural fishery as well. I think there is a little bit more leeway now because recovery is ongoing, but it is still technically illegal to harvest.
Hmm.
But recovery is going well. There are quite a few juvenile abalone out there now, which will grow up to be adult spawning abalone, and hopefully the populations will increase. Things are looking good. A lot of the monitoring programs say that there's a lot of talk about taking it off the SARA listed species list. Nothing really ever comes off the SARA listed species. We're not that optimistic, but this is a species that potentially one day maybe could. And then oysters and mussels. So no presentation for me would be complete without talking about oysters and mussels because as my mom said, this is what I study. So in BC, we have two species of oysters. We have the native Olympia oyster, and we also have an introduced Pacific oyster. So the Pacific oyster was introduced for aquaculture purposes, which is fairly evident given its size there, why they would want to do that. Nobody wants to harvest or eat hundreds of Olympia oysters to make up the meat of one Pacific oyster. It's widespread in BC now, meaning we don't really consider it an invasive species anymore. It is technically by the book definition, but it's established itself. It lives concurrently with the Olympia oyster in a lot of places, and people are just generally okay with it being there. It's not taking over the intertidal. We also have three species of blue mussels, so Mytilus trocellus, Galloprovincialis, and Edulus. And they all look very similar. And by very similar, I mean you can't tell them apart unless you do genetic analysis. They look identical. This also means that they can hybridize with each other. Oh, my lights went off in my office. I have to stand up. They can hybridize with each other. So if you are out on the beach and you see a mussel bed, depending on where you are in the coast, it's highly unlikely that this is one species of mussel, and you're more than likely looking at a mix of all the species or some mishmash of hybrids and pure species and absolutely everything under the sun. So as scientists, and what I do in my research is I just call it the Mytilus edulus complex and call it a day and don't try to do anything more than that. We have a third species of Mytilus, but it's very different looking. This is Mytilus californianus, which is the California mussel. You find it more on wave-exposed coasts. So this is western side of Vancouver Island, northern Vancouver Island, wavy places. And it's got bright orange pigment in its tissue. So before I mentioned about the Pyzaster being purple and orange, so where you find California mussels with the bright orange pigment, you can see where I'm going with this, you're more likely to find orange sea stars. So if you paddle in the Bamfield, Tofino area, you're more likely to see California mussels and orange sea stars. And if you paddle in the Burrard Inlet, more sheltered, less wave exposed areas that blue mussels really like, you're more likely to find a higher proportion of purple sea stars. So if you switch them, then they might start eating different things and they might turn different colors. That does not mean that there's only purple on one side and only orange on the other, though you do find both in either areas. Oysters and mussels are not only cool animals, they are very economically important to Canada. So oysters and mussels together account for 95% of our shellfish aquaculture in Canada, or $111.5 million or 37 tons per year. They also have a huge ecological importance. So they are filter feeders, and one oyster can filter up to 50 liters of water a day. So it removes pollutants, it improves water clarity and quality, it increases light penetration, does a bunch for the environment. They're also ecosystem engineers, and this picture is a great way of showing that. So they create habitat and they create structure for other organisms to live on. So that one Pacific oyster is housing a bunch of Olympia oysters. There's barnacles growing on the mussels. There's limpets growing on the California mussels. They create habitat for themselves. So we talked a lot about some species that you might see, and I didn't want to spend my whole talk on this. So these are the honorable mentions that I didn't have time for, but I still think are really cool species. These are moonglow anemones. They're new to me. I just found out about them last week. They're see-through anemones and they burrow themselves into the sand. So if you touch them, they retract into the sand. You have to go digging like a clam to find them. If you're in BC and on the right spot at the right time from February to April, you might see the herring spawn. So these are fish that spawn every year on eelgrass or giant kelp or bull kelp or some sort of near shore kelp. And it attracts a wide variety of sea life and wildlife. It turns the ocean turquoise blue, and it's quite a spectacle to see. Giant sea cucumbers are also a really unique species. They can regrow their internal organs. They can also eviscerate them, but then they can regrow them, so it all works out. Frilled whelks and their eggs, they come in a huge variety of colors and patterns. They look like 10 different species, but they're all the same species, just in different colors. And their eggs look like little pellets of rice. Moon snails, beautiful shells. People like to collect their shells. Not a beautiful animal, but they create those collars. I don't know if you can see my mouse. They're called sand collars, but really they are moon snail eggs. So if you come across these on the beach, the moon snail secretes a mucus and it smushes together sand and eggs, and then it protects the eggs until they're ready to hatch. They also bore little holes in shells to eat the tissue inside. So a lot of people collect the shells as well because they have a perfectly circular hole in them, and they like to use them as necklaces or as charms or something because the hole's already drilled. But if you come across a shell with a perfectly circular hole in it near where it hinges closed, that is likely a moon snail, and you're probably on a moon snail beach. The mossy chitons look very, very boring and bland, but on the inside they are bright blue, fluorescent blue. Some even glow under UV or blue light. They glow green around the edges. Sand dollars, lots of people collect sand dollars, and they're actually in the same phylum as sea stars, sea cucumbers, and sea urchins. None of those look anything alike, but they're all genetically related. Sea urchins, these ones are an interesting one. They are also related and often used as an example of keystone species. So I talked about sea stars, Pisaster as being a keystone species. Sea otters are also a keystone species. And you may have heard the example of when sea otters were overharvested and there were no more left, that the sea urchins got out of control because sea otters eat sea urchins, and when there were nothing to eat them, their populations grew drastically and they just mowed down the kelp beds. So where we used to have really beautiful, productive kelp forests, it's now just bare rock, bare sand, and sea urchins. And we call those urchin barrens. Recently, they have reintroduced sea otters to some areas of BC, and things are going really well. Sea otters also eat abalone, and there has been a decline in abalone populations in some areas that sea otters have been reintroduced, but not in all areas. So the ecosystem is finding its own equilibrium, and in some areas it's going back to the way it was before we overharvested and removed a keystone species. We're going to pivot again and talk about my research. So what do I study every day? I study ocean acidification. So the ocean is the largest carbon sink in the world, and it absorbs 30% of our atmospheric CO2. With increasing CO2 due to climate change, the ocean is absorbing more and more carbon, and it's not really going to stop. Chemical reactions then turn the CO2 in water into other things, including hydrogen ions, and this abundance of hydrogen ions decreases the pH of the water, which is ocean acidification. In turn, these hydrogen ions then bind with available carbonate ions and create bicarbonate. And that means that that carbonate ion, is no longer available to any calcifying organism. And they need they need carbonate to make their shells. So calcifying organisms are anything that builds a calcium carbonate shell. This includes snails, clams, oysters, mussels, abalone, et cetera, et cetera. They are all extremely vulnerable to ocean acidification. So my research questions aim to answer whether, one, how does OA affect the growth and survival of oysters and mussels in BC? And two, does ocean acidification affect the susceptibility of oysters and mussels to heat waves and predators? So if you're smaller or you're weaker, are you going to be more likely to die in a heat wave or more likely to be eaten by a predator? So to answer these questions, I sourced some shellfish. I found some wild blue mussels, some edulous complex, on Quadra Island, and then I bought some oysters. It's really hard to find juvenile oysters, and I wanted about 2 inches tall. So I sourced them from a hatchery on Reed Island, which is near Quadra Island. And I spent the next 2 weeks gluing individual tiny numerical tags to the outside of over 1,200 shellfish. Watched a lot of movies in that time. I had a lot of undergraduate help, but it was extremely tedious. And then I took all those organisms up to the Hackeye Institute on Quadra Island, where they have an ocean acidification lab, where you can mimic ocean acidification in a controlled environment. And I let them grow under different pH conditions for 8 weeks. So I had control oysters and control mussels, and then I had different levels of acidified oysters and mussels. So results, what did I find? How is it affecting growth? These are my control oysters. So normal pH are regular ocean pH oysters. And for context, this is what they looked like at the beginning. So everything outside that yellow line is new shell that they grew in those 8 weeks at the Hackeye Institute. Conversely, these are the acidified oysters. So they didn't put on much shell, they're much smaller, and they just didn't grow. So growth was significantly decreased by ocean acidification in my oysters. Additionally, how does it affect shells? Again, on the left, normal pH, they are more colorful, they're glossy, they put on extra shell frills, and on the right is our acidified oysters. They're eroded, they're just really unhappy looking, they're all kind of white, they're all the same color. And same goes for the mussels. So that black layer, it's called the periostracum layer, and it eventually erodes to show the blue layer underneath. So what I'm doing right now, what I did today actually was try to quantify this. I need to put some numbers to help me show what I'm seeing. And then some good news. Let's end on a good note. How is it affecting their thermal tolerance? So I wanted to see if ocean acidification and a warming climate were having compounding effects. So I'll show you one graph. This is a little bit more complicated. So on the bottom, you have temperature going from cold to hot, left to right. And then on the y-axis, up and down, you have proportional survival. So at the top is 100%. So dots that are at the top mean 100% of my organisms were alive after a heat wave. And at the bottom, you have 0%. So if the dots are at the bottom, it means that 0% of my samples were alive. And then there's going to be different colored lines. So the different colored lines correspond to my pH treatments. So if ocean acidification makes you more susceptible to heat waves, those lines will be more spread out because you will die at a lower temperature. However, that is not the case. They are all in exactly the same spot, pretty much. So I
Oh,
can say with confidence that ocean acidification is not making mussels more susceptible to heat waves. Um, Comparing and contrasting curves like this across species or across experiments is really difficult. So we use one point to describe everything, and we call it the LT50. So it's the lethal temperature at which 50% of our samples die. So for mussels, our LT50 is about 35 and a half, 36 degrees. So at 36 degrees, half of our mussels are going to be dead. And that's going to be half of our mussels, whether they're control mussels or acidified mussels. However, for oysters, it's a little different. Our LT50, so where 50% of them die, is almost 12 degrees higher at 47 degrees. So again, OA doesn't seem to be affecting their thermal tolerance. They all die at the same temperature, regardless of whether they're acidic or controlled, but their LT50 is 10 to 12 degrees higher than the mussels. So under climate change, if our climate warms so that every day we're experiencing 40-degree weather, we'll probably still have oysters and we probably won't have any mussels. But that's looking many, many, many years down the worst case scenario line. But we will say that oysters are a little bit more thermally tolerant than mussels. So this is good news. It means that ocean acidification is not compounding with temperature to make life even harder for oysters and mussels, which is great. So lastly, how can you get involved? So there are so many of you on this call and you're out on the water so often and you can observe so much more than one scientist can going out on the water once a month. So we call this citizen science and it's an extremely important part of science. Marine is the multi-agency Rocky Intertidal Network. It's a consortium of community science people, and they track many things, but one of the things they track is sea star wasting. So if you are kayaking and you see sea stars, or specifically if you see a sunflower star, they want to know. So you can use this link and you can upload a picture, you can upload an observation, and they'll use it as a data point in their monitoring programs. If you kayak a lot or you're from or you're visiting Tofino, the Strawberry Isle Marine Research Society does exactly the same thing. And at the end of the day, they give all their data to Marine anyway, but it's a small not-for-profit, which was recently bought out by Redfish Restoration, which is a great organization. Both of them take volunteers. Redfish does aquatic restoration across the board, freshwater and saltwater, and they're always looking for volunteers to help with some of their community projects. So if you're ever in the area, these are both great things to look up. iNaturalist is my social media of choice. It's an ID app that you can use. You can upload something you see, a picture of something you see in the field, and other users can ID it for you. It's also used for a lot of citizen science across the board. There is, for instance, if you're kayaking or hiking in a BC provincial park, they want to know what organisms you find in all BC provincial parks. So they have projects. So you can upload your observation to a project. And one of those projects, in addition to the BC Parks project, is the Tracking Starfish Wasting and Recovery, and it's led by the California Academy of Science. So if you don't want to upload your observation to Marine and you don't want to volunteer with Strawberry Isle, that's fine. You can upload your observation to iNaturalist from your couch, and then somebody from the California Academy of Science will add your observation to their database without you having to do anything extra. So I did this the other day. I didn't even know it was a thing. I uploaded a picture of a piesaster and then I got a little ding that somebody had added it to the database. So that's a really easy way to get involved. If sea stars aren't your thing, OceanWise has a marine monitoring program for whales. If you're kayaking and you see whales or porpoises, then you can let them know and they'll broadcast it to commercial vessels like tankers. So there are fewer collisions. We are currently in Canada Ocean Week. It's a national celebration of the ocean from coast to coast to coast, and it culminates on June 8th with World Ocean Day. So there are so many ways you can get involved with this. Check out Ocean Week can.ca for events. They're happening across the country, um, on the coast, obviously. And I will plug Tara's event, which is happening on June 8th at Leg and Boot Plaza. So if you're in Vancouver on the seawall, um, Tara is our undergraduate outreach coordinator and ocean acidification education assistant, which is a very long title. And she's hosted um, this event. I believe Chris Harley will be there. It's just about promoting False Creek Friends and the intertidal life and marine life in False Creek. There's also bio blitzes that happen every year. This was a poster for the 2022 bio blitz that happened in False Creek, but they happen every year. A bio blitz is where they use citizens to go out and just catalog and inventory every single species that you find in a given area. So this bio blitz was in False Creek. Last year it was on Quadra. This year they're having a bio blitz for World Ocean Day. It's on June 8th. It's with the Stanley Park Ecological Society, the Vancouver Aquarium, and the Water Rangers. So they're celebrating World Ocean Day with a bio blitz in Stanley Park. So everyone is welcome to that. And there'll be people there to help you ID and to tell you even more about the cool things you're seeing. If none of this is interesting to you, you can check out the Government of Canada has a citizen science portal. So everything I've talked about is marine related, but the citizen science portal has birds, bees, viruses, belugas, pretty much anything you could think of. If you want to get involved in science in any way, shape, or form, you can check out their portal. That's all from me. That's it. Thank you so much.